Hello friends, today we are going to discuss about fermented beverages, especially beer and wine. Fermented beverages means the incorporation of a microbial species to generate a drink which is refreshing at times and most often do have some additional nutritional properties. There are so many to name, but we are mostly concerned here with the alcoholic beverages which contains approximately 3 to 40% of ethanol in it. There are several types of these alcoholic beverages, but can be broadly classified into two categories. Some produced only by fermentation, while some are produced by fermentation and then followed by a distillation step. This distillation step gives higher ethanol content because we are going to purify the product, and as such the ethanol percentages increases to approximately about 40%. Let us see some examples. The ones which are produced solely by fermentation and are not distilled are beer and wine, while on the other hand, the ones which are produced by distillation are known as hard drinks or liquors, at times even go by the name of spirits. And some examples include whiskey, vodka and brandy. Because we are discussing here mainly the only fermented product, that is beer and wine, we will also lay stress upon the organism which is going to produce it. And in most cases, we commonly hear the name brewer's yeast, which is nothing but Saccharomyces cerevisiae. These are budding yeast. And what do? They generally utilize glucose and convert it into ethanol and CO2. In a fermented drink, this ethanol content gives the typical flavor and aroma as well as taste, which is unique of that kind of a beverage. We will first begin our discussion with beer. Beer is a very common fermented drink used or consumed worldwide, and interestingly, it is the third common most drink all over the world. First is water and then tea. Next comes beer. What is this beer? It is actually a fermented drink produced from several grains, most importantly barley. History also shows that there are several use and consumption of these fermented beverages, and we can also see that in the medieval age some German people had the consumption of beer, which is pictorially represented here, and we do have documents of several other such in the history. Let us see what are the different types of beer first. There are so many to name. Mostly the names are derived from the location where they originate. Now, common ones are lager beer, ale beer, porter and stout, and these have different body. As you can see, by body we mean the color and the texture. Now basically mostly consumption is of the lager beer and ale is lesser used. The ale beer are preferred only in regions and they have slight more bitterness, while porter and stout have full body and some are even rich. What are the ingredients which are used in beer making? Firstly, the barley, which should be malted. Then we have the hops. Without hops, there is no beer. And then we have the fermenting yeast, which is Saccharomyces cerevisiae. We'll go one by one, but before that, we will look into the complete picture of the steps which generates the beer. The barley is first produced into malt. Then the malt is milled. Then this goes into the mashing tun, which causes a mashing. We will discuss mashing in detail. Then there is a lautering and final filtration. The filtered wort is taken into a copper kind of a container where it is boiled with hops, a plant product. Then there is a separation and cooling of the product. Then that is sent to the fermentation tank. In the fermentation tank, yeasts are added and left on for the required time period, during which the yeasts will grow and we can have the extra yeasts taken out. After the fermentation is over, the beer is sent for maturation in a maturating tank, which is then followed by certain pasteurization and filtration steps, and then goes for the packaging into either bottles, cans or kegs. Let us see with the starting material that is barley. Barley should be of typically good variety, meaning microbiologically and bacteriologically sound, and most importantly, it should have a good germination index. The barley required for beer production should germinate at least 97% within 5 days. The barley that will be used for production of beer is generally stored in the silos and then it is used as required. In the malting step, it consists of 3 different stages. The first is known as steeping, which means that it is soaked in water. And then it is taken to the malting bed. We can see here a malting bed where the germination takes place. Finally, it is taken to a kiln where it is dried. In more details, so the steeping basically actually enlarges the barley grain. So it's all to do with the barley grain. We are first allowing the water seeping, which allows for germination, which we can see here with rootlets coming out. It is germinating, and we are not allowing complete germination. Instead, we are partially germinating and then arresting the germination by kilning, which means the incorporation of heat. This also dries the barley. Now this is ready and we call it malt. Let us see in a barley grain which are the regions important for us. We have the aleurone region here as well as the scutellum region here. These two are the live membranes, and they are important because they generate and produce a host of enzymes and hormones which will degrade the endosperm containing starch and proteins, which are the food material for the barley grain. Now these food material will be broken down by the enzymes and the hormones which will generate food for the yeast. We don't want the grain to grow into a plant, rather we are stopping the complete germination and allowing the degradation of the complex starches and proteins and seeing that they are provided to the yeast, which we are going to inoculate so that they get the food and convert it into ethanol. After the malt is ready, here actually we are seeing the milling. That means the malt, the dried barley grains are made into thin and fine powders. That is milling. Once milling is done, that is taken for the mashing, and we will have to see elaborately what mashing does. And this is the filtration and wort boiling step. In the mashing, we have to actually undergo and see what biochemistry is involved. Mashing means the incorporation of hot water into the ground material that comes after the malt. This hot water has several roles to play. The temperature of the water is important for the degradation of certain components, again in the barley grain, that is cell wall degradation, then followed by protein degradation. As we increase the temperature, then we get the degradation of the starch. First, the starch gelatinizes and then saccharification. That is starch, which is a macromolecule composed of several molecules of glucose in a polymeric composition that is broken down into free glucose units. Yeasts can take up these glucose units and cannot degrade starch at the first place. Mashing generates a liquid which is known as wort. Sometimes we call it sweet wort, meaning all the starches have been degraded mostly into glucose and maltose. Now these two components will be the food material for yeast. But right after the production of wort, we have another important step in beer making, and that is the wort boiling step, where incorporation of hops are important. We can see here hops are being added to the wort which is being boiled in a copper kettle. What are hops? The hops are actually the dried female flower cones of the plant Humulus lupulus. And if we actually dissect, we will see that these clones have lupulin glands almost towards the base of the cone. These lupulin glands contains resins and essential oils. What are the important components of these lupulins which are so interesting, and why are they added in beer? There are some substances in these resins and essential oils which are called as hop acids. And interesting to note, hop acids are bitter. Now we have two such components here, principally alpha acids and beta acids. Here shown are the structures of the two. This is alpha acid, this is beta acid. They have several other derivatives as well. The alpha acids are known as hemulunes and beta acids are known as lipulones. Other derivatives are common, but let us concentrate only on one to understand what happens during the wort boiling step. During the boiling, here you can see the arrow with the temperature direction. These hemulunes or the alpha acids are converted into their cis and trans isoforms. That means they are converted to their isoforms, and these isoforms are even more bitter. So beer, all those who have tasted knows that it has a bitter taste, and this bitter taste comes definitely from the hops and principally these kinds of essential oils. Now, after we have boiled with the hop, as we have shown in the previous diagram as well, they are cooled down, the particulate matters are separated, and then the whole thing is filtered and goes to the fermentation unit. In the fermentation chamber, the yeasts are added, which carries on the fermentation to approximately 48 hours or even slightly more, and during which ethanol is produced as well as CO2. The CO2 gets out in the form of bubbles, as we can see the frothing here actually. And after the fermentation is over, it is sent to the maturation tank. What happens in a maturation tank? Basically, the temperatures provided in case of beer production are obviously the growth range temperature of the yeast. That is somewhere near 30 degrees, 25 or 30 degrees centigrade. But beer often is consumed chilled, and that is the reason we send it to the maturation tank and give a lower temperature, the consumption temperature of beer. Why we carry out this step is because by chance if there is a colloid formation in the lower temperature ranges, then they can be separated out. Now, if we can look into this diagram, there are two glasses of beer. One, this is actually an unstable beer where haze is formed, meaning there is haziness here. While if you can look into this one, this is a colloidally stable beer, means there is no colloid formation. What happens is there are certain components in barley, more in the hops, which reacts with the proteins present in the mixture to give rise to a colloid, and these colloids forms as particles and they cause this haziness. Nobody will like to have a hazy beer, and as such clear part or transparent kind of nature of beer is essential, and so that is checked by keeping it in a lower temperature because colloids will be formed only in the lower temperature. So once maturation is done, it is kept for even some more time, and then we see that the yeasts will settle down and then can be easily removed. These are the structures of certain formulations like polyproline and polyvinyl polyproline. These two are analogues, and often during the maturation stages these can be added so that the colloids, even if they are getting formed, they will be arrested and can be removed by filtration, so that once it is bottled there is no colloid formation after the bottling step. After it is produced, it goes for the packaging unit where the filling first happens into the respective bottles, and then it can be even corked or required type of labeling can be done. But one more essential step here is the pasteurization step. Most beers are pasteurized, some by tunnel pasteurization and some by flash pasteurization. And because packagings are different, they are all sent in conveyor belts and accordingly packaged. Some are bottled, as we can see here, some are kegged. Kegs are larger units which holds a huge amount of beer, and whenever required, the tap can be opened and you can consume. Or you can also pack beers in cans. So we have basically seen the process. Now there are certain qualities of beer which are important and to note. Foam. Beer without foam is not to be seen. So how exactly this foam is produced, or what causes the formation of this foam? There is a typical compound elaborated as lipid transfer protein 1, which is most important, but there are other compounds along with this which generates the foam, and obviously that means this lipid transfer protein 1 comes from several ingredients which are from barley or from the hop material that is present in the mix. Flavors of beer are different, and that varies from the organism that we have used, because although yeast, the strains can vary, and like Carlsberg uses a typical strain known as Saccharomyces cerevisiae carlsbergensis. So the carlsbergensis strain will have a different kind of flavor and aroma giving compound and may differ from other beer types. That is why we have ale and lager separately. While the aroma compounds are again generated during the maturation stages, when after the complete fermentation step, the yeast still remain in suspended manner and they generate some other aroma giving compounds, and like higher alcohols, esters, and some other aldehydes and ketones. So this is what generates beer. So we have seen a whole lot of study about the beer, how it is produced, the starting material, and then final packaging and sale. Now there are so many more to mention, but we will then continue with the wine now. Wines basically are the end product of complete or partial fermentation of the juice of grapes. It is important to note that it must be grapes, and commercially speaking, the wine that is produced should come from the grape variety which is known as Vitis vinifera. You can see the black grapes here. The wine grapes, let us briefly see what they are made of. Only then we will understand what is the actual substrate which is being utilized by the yeasts here. It has a lot of substances. The grape on the skin has lot of potassium, mineral salts, oxidases, that is enzymes, primary aromas and astringency compounds, while it lacks sugar and acidity, which is found mainly in the central and the epidermal regions here. So, grape is very important, and there are several cultivars of this, belongs to the family Vitaceae and genus is Vitis. We'll move on to the types of wine next, where we have several on line. Let us first see the red wine, which means obviously red in color but produced from red grapes. And white wine, is interesting to note, can also be produced from red grapes and most often is done so, although the color is white. Sparkling wines are wines which has the excess CO2 in it. So when they are poured, you have the generation of this sparkle. Champagne, I think all of us know of, is example of a sparkling wine. While still wines are somewhat like this, which do not have any CO2 as effervescence here. Dry wines basically means that there is no sweetness of the wine and most of the sugars have been completely fermented. Sweet wines means some sugars are left and it is sweet to taste, while fortified wines are where the ethanol content of a wine is increased by addition of simply ethanol. We will briefly go through a flow chart which gives us the idea how wines can be produced in the industries. First, they are sampled, that is in a vineyard, that is checked and then harvested, means picked. Then the grape that is procured is processed and then converted into must. Must is actually sulfited to reduce the load of unwanted and contaminating microbes. Then as required, it can be centrifuged, filtered, sedimentation can be brought about or clarification. Then it is heated and cooled. Finally fermented with yeasts. Then other steps after fermentation includes racking, then blending if required, funnel clarification, filtration. Aging is important in wine. Then wine can be analyzed. If there is some changes, final filtration can take place, then bottling, packaging and sale. In the sampling part, it is important to note that there is a small instrument called as hydrometer which can be held just beneath the grape and seen to the sugar concentration. Ripeness is very important. It should not be over or underripe. Harvesting can be done by hand picking methods or can be done by machines. Machines generate faster but carries on leaves and other herbaceous material, so wine may have a taste of herbaceous nature if they are mechanically picked. Then in the following stages of processing, the grapes are sorted. Sometimes they are also cleaned, weighed, and all that happens on a conveyor belt that carries on to a destemmer to remove the stems. This is a destemming machine. We generally go by the name of stemming, where stems are being removed, and the same machine also crushes the grape and generates the liquid which is known as must. We can see here the mixing or the conversion of the grape into must. It is done by hand, but a machine can also do the same thing. So this is of white grape, this is of red grape. Then moving into the next stages is wine press. Now here it is important to note that the two wines, red and white, the major varieties, are produced by slightly different processes. The initial part is almost the same, but here after the must is produced, if we are heading towards production of white wine, there will be a press which will take out most of the juices and leave on the skins, while for red wine there is no requirement of a press. In case of early times, this is a traditional basket press and this is a horizontal newer technology press. Clarifier is again an instrument which clarifies, that is, removes any suspended material that may be present in the must. But before moving into the fermentation, we will definitely have to carry on steam cleaning and sterilization of the fermenter which we are going for fermentation. In the fermentation stages, the yeasts are important. Wild yeasts are so many to name, like Candida, Hansenívola and Cloacara, but mostly they are not used because they may not give the desired end product all the time. And so we follow wine yeast, and importantly Saccharomyces ellipsoides are the ones which are used for wine making. In the fermentation stages, they are kept for the required time period, followed by temperature requirement, and then we get a final product. White wine requires more time and lesser temperature. Red wine requires higher temperature and lesser time. After that, they are again sent to presses after the fermentation to remove any suspended matter. And finally, we carry out a malolactic reaction or fermentation, which is the incorporation of malolactic acid bacteria. Some examples are lactobacillus, oinococcus, pediococcus and leuconostoc. Here is shown the oinococcus species, typically meant for the mostly used malolactic converter. What it does, it converts the malic acid present in the grapes to lactic acid and carbon dioxide. Now, why do we need to convert the malic acid? Because malic acid, if not converted and the amount is high in the produced wine, it will cause some kind of an astringency and sharpness, while lactic acid has a smoother feel. After that, the analysis is carried on. You can see here analysis of the sampling port from these are undertaken, and then followed by bottling. Now, in many cases, the bottling is done later and there can be an aging step in between, while in many others, there is a aging done before in large oak barrels followed by bottling. So, it depends on the producer. So, bottling obviously will be followed by corking. You can see here the corking stages. You can see here the corking instruments going around for corking of the bottles, and aging obviously maybe after this or before this, most often before this. Oak barrels do have important roles to play in the generation of some of the flavors. Now, we have seen some of the ways how wine can be produced, whether red or white, but we will end up our discussion with knowing there can be several wine defects. And to name a few, there can be some which can be caused by the yeasts, and there can be some which can be caused by bacteria. To list down, what basically happens is, mostly if yeasts remain within the bottles even after the final filtration clarification stages, they may carry on further fermentation, which will generate more of ethanol and CO2, which may not be required by the specific type of wine. Like we have Saccharomyces cerevisiae, Bionas and Bailey, they generally cause this problem. While Saccharomyces ludwigi and Brettanomyces will produce higher concentration of ethyl acetate, and these are having several other types of flavor not desirable in wine. Then we can have some film forming yeasts by the name Hansenívola candida. They form a film-like substance on the surface of the wine, which is obviously undesirable. And then we can have bacteria like lactic acid bacteria and acetic acid bacteria. These will produce acetic acid, lactic acid, and obviously lower the pH of the final product, which is again undesirable. So much so, we have seen how wine can be produced, but we will end up by giving you a list of all these 41 different aroma and flavor giving compounds, so that we can always remember that, let us see one of them, methyl butanol, ethyl caproate, or methanol, citronol. So many to name. There are so many flavor giving components in wine, and not to forget, red wine has antioxidant effect and property, so we can consume obviously in lower levels to have some good taste and feel, as well as have some beneficial effects as well.